Hello, my lovely anatomists and physiologists. Michelle Glass here with our chapter 14. And this is our last chapter for anatomy and physiology part 1. Yay! All right. The topic for chapter 14 is all about the somatic division of the nervous system, which is, of course, the division that's controlling our skeletal muscle, right? So we're thinking here of our body in terms of our skin and our musculature and to some extent the skeleton, right? Because the muscles attach there. And so let's start with this idea of the nervous system, remember, as a communicator. And so the beginning part of quality communication is listening, right? Collecting data, collecting information. And so we're starting here with sensation. When we talk about sensation, what we're talking about is the activation of sensory receptor cells at the level of the stimulus. So this is kind of our definition for sensation. And we're going to spend a little bit more time here picking out what we mean by receptor cells. Before we move into that, though, let's take just a minute to talk about this term perception, which is slightly different. In the case of perception, this is the central processing of the sensory stimuli. Into a meaningful pattern. And this is the thing that's very interesting about our nervous system, is pattern oriented, right? So we've talked about this sort of idea of the same information is traveling the same routes all the time. And because we've been experiencing that for our whole life, we begin to have an understanding of that sensation as pain or as a nice touch or a bad touch, right? And so we begin to perceive that sensation, right? So the perception is talking more about the processing part, and the sensation is talking more about stimulating basically those receptor cells. So let's pick up on that just a little bit more. When we are talking about our receptors, we can talk about these as cells or structures that detect sensation. And there's a variety of sensations that our body can detect. So we'll be talking about all of those in just a little bit. Very often when we're talking about our receptors, we're talking about what we can call transmembrane receptors. Trans means across, and membrane is, of course, referring to that cell membrane. And so transmembrane receptors, these are basically proteins embedded in the cell membrane. This is all taking us back to what we talked about in our chapter 3. When we talk about these transmembrane receptors, very often what we're going to see is that they're actually ion channels, and very often the stimulation means to open ion channels. When you open ion channels, we should think mostly this is talking about depolarization. Depolarization, remember, is the movement of a positive charge into the cell, making the cell more positive. That's a stimulation, right? We also can see that these transmembrane receptors, these special proteins, can cause changes to this cell signaling process. And we're going to get more in depth on this conversation, this part of the conversation, in Anatomy and Physiology Part 2. So we'll see things that involve those G proteins and second messengers, which we've mentioned just a little bit about this semester, but we'll get into more detail in the future. When we talk about these receptors being stimulated, what we see very often is that they can be activated by ligands. So we've talked about a ligand gated channel as the chemically gated sodium channel that our neurotransmitter acetylcholine bound to, right? And so ligands can be neurotransmitters. We'll also see that ligands can be something like a chemical in food. So when we talk about gustation or our sense of taste, it's different chemicals from our food that are binding to these ligands and stimulating those receptors. Or in the case of smell. So we can also talk about like chemicals in air, right? And so we'll see olfaction is working in a similar way. We'll also see that receptors can be stimulated by changes in temperature. We can have mechanical activation. So we're going to see this in touch. We're going to see this, and here let's use touch in a broad sense. So let's think about this in terms of pressure, vibration, those sensations we talked about, like itch and tickle. We'll see this in hearing. And we'll see it in also, I'm going to put it here, equilibrium and balance. And we'll come back to this idea more in the future. We can also see our receptors activated by light. So this is part of what we're seeing in the eyeball with our special photoreceptors. So we'll talk more about those. When we talk about these receptors, we see that they can be classified based on cell type. So we have what are described as free nerve endings. We have what's called an encapsulated ending. And then we have what's called a specialized receptor cell, where I think we can think about the free nerve ending as the most simplistic version, and then becoming more and more specialized, as the name implies. And specialization here is talking about like particularity. It has like a very specific goal, right? When we talk about a free nerve ending, all we're talking about are dendrites embedded in tissue. And we see that this is how our pain receptors and our temperature receptors are functioning. So this is the most simplistic. You just have that dendrite, that part of the sensory neuron that's collecting information, and embedded in the membrane of the dendrite, you have receptors for pain, receptors for temperature. In the case of the encapsulated ending, it's the same kind of idea, but it's covered. So the dendrites are covered with connective tissue. And this is going to increase the sensitivity. And here we're going to see this with pressure, and we're going to see this with touch. We talked about some cells in the integumentary system, and we talked about them as being corpuscles. And that's really, I think, what we're talking about here. When we talk about the specialized receptor cell, this is going to be having distinct structural components. So the anatomy here in the cell is significantly more complex. These specific structural components or distinct structural components help to interpret a specific type of stimulus. And I'm just going to put here, we'll see this in our special senses, and we're going to define what we mean by special senses here in just a few minutes. You kind of know this. This is those special senses that we think about from an early age. So like vision, taste, smell, hearing, and even what we talk about is equilibrium and balance. Touch is actually not a special sense. So touch, remember, is part of that encapsulating ending type of receptor. Okay, when we talk about receptors, we can also talk about them in terms of their classification by position. And so here we have three. We have what's called the exteroceptor. We have what's called the interoceptor. And then we have what's called the proprioceptor. Where the exteroceptor, think external to the body, and so these are positioned near external environment. Which I'm struggling here, so let me rewrite the word environment. So exteroceptors are helping to collect information about our environment. So even vision, even taste, smell, pain, temperature, touch, all of these would be classified as exteroceptors. So these are going to really be those sensations we're consciously aware of, because it's important for us to collect information about our environment to keep ourselves safe. Interoceptors, then these are going to be more internal. So this is internal organs and tissues. So here you should think about this more in terms of the autonomic nervous system. So collecting information about blood pressure and maybe internal body temperature, pH of your blood, your gases in your blood, dissolved gases, right? And the proprioceptor is going to be detecting that sensation of proprioception, which remember is talking about the position of our body in space. And so here you're going to find these cells in moving body parts. So here, think joints, think tendons, think ligaments. Okay. The last thing that we want to do here is talk about, we're going to go back and forth. So let's look again at our discussion of sensation, and let's divide this into what we can call general senses and special senses. And let's actually define our special senses first. Here you're going to have a specific organ, and you're going to have one of those specialized receptor cells. Receptor cells. These are going to be the sensations of gustation, which is taste, olfaction, which is smell, vision, hearing, gravity and acceleration. And I'm going to say position of the head in space. Now, this is all discussed in our Anatomy and Physiology Part 2 course. So when you're reading your OpenStax chapter 14, you'll see it goes into all the details of these special senses. We will do that in the future. So you may skip that for this particular chapter. So anytime it's talking details about these special senses, just skip over that and save that for Anatomy Part 2. When we're talking about general senses, this is where we're talking about free nerve endings. or where we're talking about those encapsulated nerve endings. So these are more widespread throughout the body. We have these receptors just embedded in structures of other organs. So we talked a lot about this in like our integumentary system. So these are going to be touch, which can be either fine touch or crude touch, pressure, vibration, stretch, hair follicle placement, where you can use the term position, proprioception, remember the position of our body in space, kinesthesia, which is that feeling of our body moving, the movement of our body. Pain and temperature can also be listed here. And here we're talking about temperature sensation that we're detecting with our integumentary system. Let's go ahead and remind ourselves of some specific receptor cells. Okay, so let's talk about some specific receptor cells and some specific type of receptor cells. So let's actually start with some types first. So we talked about classifying receptors based on cell type. We talked about classifying them. Let me take our screen down. We talked about classifying our receptors in terms of cell type. And we talked about position, but we're also going to see we can do it based on function. So I'm going to make a little bit of room here to talk about our different functional descriptions. Okay, so we can have what's called a chemoreceptor. Chemoreceptors are going to respond to chemicals. So we're going to see this for gustation, taste, olfaction, sense of smell. We can have osmoreceptors. This is detecting difference in the solute concentration. Remember, solute is the stuff that's dissolved in the water. So this is talking about how salty are your fluids. We have a receptor that's responding to pain. We call it a nociceptor. We have mechanoreceptors. These are going to have kind of like a physical change, right? These maybe have mechanically gated ion channels. And so these are going to be our sensations of touch, pressure, vibration, sound, body position, head position. We also have thermoceptors. These are detecting differences in temperature. And we also have baroreceptors. These are detecting differences in pressure. Maybe let's identify here in fluid. So when we're talking about like changes in blood pressure or buildup of pressure inside the urinary bladder, then we might be talking about baroreceptors in that case. Let's go back here and then mention some specific receptors that we've talked about in previous lectures. So we've talked about Merkel cells. These are all things we talked about when we did our integumentary system. So Merkel cells are going to be involved in detecting vibration and touch. We have the Pacinian corpuscle, which is going to be involved in detecting vibration also. We have Meissner. Corpuscles, which is detecting light touch. We looked at the hair root plexus at the base of each one of those hair follicles. So this is going to be detecting movement of the hair. Okay, so these are all some examples, some specific examples of receptors. All right, that's it for our discussion here of sensation. Stay tuned for our next video, looking a little bit more detail at our spinal nerves. As always, take care of yourselves and each other.